Well, thank you. I'm going to talk about targeted capture and massively parallel sequencing for genetic testing of breast and ovarian cancer. And the overview, I'll start with the current approaches for genetic testing of inherited breast and ovarian cancer, and then describe how we developed and validated a targeted capture massively parallel sequencing approach that we call BROCA. And then take you through our data on the first clinical application of BROCA, which was to study 360 unselected ovarian cancer patients that had their surgery performed here at UW. So BRCA1 is a highly penetrant breast cancer susceptibility gene. It was originally identified by studying linkage in severely affected families. This is family 1 from Mary Claire King's series, which she published in 1990. And in this pedigree, breast cancer patients, females are indicated by filled circles, and their age of diagnosis is indicated underneath the filled circles. So in this case, this woman was diagnosed with breast cancer at age 59. And as you can see in this pedigree, there were lots of cases across 4 generations. Breast cancer is inherited dominantly. And this family is characterized by very early onset of breast cancer. There are 5 cases of breast cancer diagnosed in early 20s, and many in the 30s and 40s. And using this family and other families in her series, she was able to determine that the critical gene for breast cancer susceptibility in this and other families mapped to a region on chromosome 17q21. But as you can imagine, back in 1990, without any genome sequencing or any even draft of the genome sequence, it was very hard to work out where the gene was on chromosome 17. So it took about 4 years to pinpoint the gene, which she named BRCA1. And after it was identified, researchers went back to their families and sequenced the coding regions of BRCA1 to look for mutations associated with breast and ovarian cancer. And in family 1, there was a 2 base pair deletion at position 2800 that leads to a frame shift and the generation of a truncated protein and a protein with aberrant function. And in this, the mutation is indicated by a red V for variant. And you can see that almost all the women with breast or ovarian cancer inherited the 2 base pair deletion. There are a few exceptions, including this woman who was diagnosed with breast cancer at age 45, but doesn't have the mutation, the BRCA1 mutation. And although it's a very high-risk mutation and a high-risk gene, there are cases within families where a woman will carry a deleterious mutation of BRCA1, like this woman here, who's now 81, but hasn't developed breast cancer. So it's certainly penetrant, but not at 100% penetrant. And BRCA1 is a classic tumor suppressor gene in that it requires 2 hits to inactivate it. The first is generally inherited in the germline, and the second occurs somatically in the tumor, and it's generally a large chromosomal rearrangement or deletion. So BRCA2 was its second breast cancer susceptibility gene, was identified 2 years later in 1996. And at this point, genomic sequencing had begun to take off. And in fact, the Sanger Center in the UK was responsible for sequencing chromosome 13, where BRCA2 mapped. So the process of identifying BRCA2 was a lot quicker than BRCA1. BRCA2 is different than BRCA1 in that it also predisposes to male breast cancer. So this is family 16 from Marie Claire's series with a 4 base pair deletion in BRCA2. And you can see there are 4 male cases of breast cancer, each with a variant in this family. So looking at the penetrance of the genes in a population-based series, Marie Claire and colleagues looked at more than 1,000 Jewish women with breast cancer in 2003. And by directly genotyping for the Jewish mutations and testing families, relatives, and unaffected members, they were able to show that the lifetime risk for developing breast cancer for BRCA1 or BRCA2 mutation carriers is close to 80%. Using a similar approach, Finch et al. In 2006 looked at the risk of ovarian cancer. And you can see it is lower. It's about a 50 to 60% lifetime risk for BRCA1 mutation carriers, and then about 25 to 30% for BRCA2 mutation carriers. So about 10,000 new cases of breast cancer are diagnosed with BRCA1 and BRCA2 mutations, and there are now a number of clinical recommendations for women who have these mutations. And they include, of course, annual mammogram, but also supplemented with more sensitive MRI screening. Patients are recommended to undergo risk-reducing surgery to remove the ovaries upon childbearing. This obviously protects against ovarian cancer, but also protects quite a lot against breast cancer development. Patients are also encouraged to discuss the option of risk-reducing mastectomy and then consider chemo prevention options. So the last couple of years, there's been quite a bit of excitement with the advent of PARP inhibitors. These are drugs that seem to act preferentially on BRCA1 and BRCA2 mutant tumors. So there is a potential to tailor a patient's chemotherapy to a particular pathway. So PARP is an enzyme that repairs DNA via the base excision pathway. If you inhibit PARP, you get specific DNA lesions that can no longer that require functional BRCA1 and BRCA2 to be repaired. So obviously, tumors lacking BRCA1 and BRCA2 are sensitive to PARP inhibitors. And there are a number of PARP inhibitors in clinical trials right now. And this, of course, has increased the clinical incentive to identify women who harbor these mutations at an early age. So BRCA1 and BRCA2 are actually very large genes. They both span about 100 kb of genomic sequence. Looking just at coding regions, BRCA1 spans about 5 kilobases, and BRCA2 is about twice the size, 11 kb. And as you can see from this NHGRI plot, the mutations are pretty much scattered throughout the gene. There are exceptions in specific founder populations, like the 185 del AG in the Ashkenazi population, and a 5 base pair deletion of BRCA2 within Icelandic population. But in general, the mutations are quite rare, if not private, and are scattered throughout the gene. So you really need to fully sequence the gene in order to be confident that you've detected all mutations. So in a study we published in 2006, we showed that amongst the mutation spectrum of BRCA1 and BRCA2, there are exonic deletions and duplications, which have now become known as copy number variants, or CNVs. And BRCA1 is particularly susceptible to these mutations because it's a very genomically unstable region. About 50% of the 100 kb locus is comprised of ALU repeats, which are highly susceptible to crossing over events during replication. So exons can be looped in or looped out, and they in turn cause either deletions or duplications. So amongst BRCA1, the frequency of CNVs is about 10% of all mutations. It's a lot lower in BRCA2 because it has a lot less ALU sequence. But unfortunately, CNVs of this type can't be detected by standard PCR and sequencing, because you can put a primer in a deletion or a duplication, you'll still get a product, but based on the sequence, you can't determine whether or not you've got 2 copies or 1 or 3. It's just not quantitative. So in the US, the current genetic testing of BRCA1 and BRCA2 is performed exclusively by Myriad Genetics. They have the patent for both genes. The patents are currently under judicial review, and I think most people believe it will probably end up in the Supreme Court by 2012. But the patent does expire in 2014. So Myriad's protocol is based on standard PCR amplification and Sanger sequencing on capillary instruments. After our 2006 paper, they did add a supplementary test, which they called BART, and it detects CNVs at exons by doing a copy number analysis at each exon. And the testing of the other genes, which I'm going to talk about, is generally done on a gene-by-gene basis in academic and non-profit labs. And most of these other cancer genes are not patented. The one exception is PALB2, which is also patented by Myriad. So I think it's become clear as more people have got familiar with next-gen or massively parallel sequencing that there's an increasing discrepancy between the cost of these new sequencing approaches and the current very high cost of the clinical gene-by-gene testing. And we believe it's not economically sustainable to keep testing sequentially gene by gene when these technologies are available, which are much cheaper and much more high throughput. So our goal was to develop a single comprehensive assay, and we wanted to be able to simultaneously screen all known breast and ovarian cancer susceptibility genes, and at the same time detect all classes of mutations, so small and large mutations in one go. So looking beyond BRCA1 and BRCA2, what are the other genes that predispose to breast cancer development? The first class are these rare cancer syndromes, such as Li-Fraumeni, caused by germline mutations in P53, Cowden's disease, caused by PTEN, diffuse gastric cancer, which predisposes to a histological subtype called lobular carcinoma, caused by germline mutations in the E-cadherin gene, and also Peutz-Jeghers, which is caused by germline mutations in the serine threonine kinase 11. And because we're also interested in ovarian cancer, we also looked at the mismatch repair genes, MSH2, MLH1, PMS2, and MSH6, because they also predispose to ovarian cancer within the context of Lynch syndrome. So the second class of breast cancer genes are these so-called intermediate or moderate risk, and they've been identified by resequencing studies over the last 5 years. People have basically taken candidate genes that either interact or are in the same pathway as BRCA1 and sequenced them in families that don't have mutations in BRCA1 and BRCA2. And over the last 5 years, there are at least 9 of these that have been identified now. ATM, BRIP1, PALB2, and more recently, RAD51C. And as you can see from this cartoon, all these genes do in fact complex in the same pathway as BRCA1 and BRCA2. And it's become known as the BRCA, Fanconi, or homologous recombination repair pathway. And we now know from a number of studies that mutations in any of the 9 genes in blue give rise to about a 2- to 4-fold increased risk of developing breast cancer for patients that harbor germline truncating mutations in these genes. So it's definitely lower than the BRCA1 and BRCA2 risk, but it still represents greater than 25% lifetime risk for women who carry these mutations. And so it's considered a clinically relevant level of risk. And we believe that women who carry mutations in these genes would greatly benefit from increased surveillance and screening. So the targeted capture massively parallel sequencing approach is going to capture 21 genes from these 4 categories, the high-risk genes, of course, BRCA1 and BRCA2, the moderate-risk genes from the cartoon, the genes within the rare syndromes, and then the Lynch syndrome genes. And we also want to be able to detect all mutation classes, so the single-base substitutions, such as nonsense, splice sites, and missenses, and of course insertions and deletions that lead to frame shifts. But also we want to be able to detect exon duplications and deletions, so we don't have to do that second test. So we call that test BROCA in honor of Paul Broca, who was a French 19th century neurosurgeon and anatomist, and he was one of the first to actually describe high-risk breast and ovarian cancer families. He published his pedigree with a very severely affected family with breast and uterine cancer in 1896. So the first step before sequencing is obviously to enrich the region you care about. And the standard approach at the moment is just to do PCR. So you basically, in this case, you would, if you cared about an exon of BRCA1, you would put flanking primers covering the exon and adjacent splice sites and generate a product by PCR and sequence it. What we've done is used complementary RNA baits from Agilent, and we use these to enrich through hybridization. So this is the same BRCA1 exon, and here are the baits, the cRNA baits, centered on the exon, covered with a 3x tiling strategy. So each one overlaps by 40 bases, and then marches into the surrounding intronic sequence until it hits a repeat region. And we obviously try and avoid these because they're a very high copy throughout the genome. And if we targeted these, you'd be pulling down basically the whole repeated portion of the genome. So in addition to exons, we capture flanking introns, untranslated regions, and we go plus or minus 10 kb upstream and downstream to look for potential promoter or enhancer mutations. So the baits are actually biotinylated. So after hybridization to your DNA library, you can pull apart the hybrids with a magnetic bead that's attached to a streptavidin molecule. And of course, our design file is not patented, and we make it freely available to anyone who requests it. So the sequencing procedure is outlined here. We start off with DNA extracted from blood, about 3 micrograms. We sonicate it down to about 200 base pairs. We then ligate on Illumina-specific adapters to make a library. do the hybridization for 24 hours that enriches for the 21 gene regions that we care about. And then after that step, we do a bunch of stringency washes to wash away the non-specific binding, and then do some PCR. And at this step, we incorporate unique 6-base pair barcodes. And we have 69 of these at present that have been validated. So you can actually multiplex the samples in a 96-well format at the moment. Once the libraries are quantified, we then pool them with equal amounts and then load them on a single lane of a high-seq flow cell where they're bound to complementary primers attached to the glass slide, which are complementary to the adapters that were ligated on the first part of the library prep. We then run the instrument for 10 days. It's a very long experiment, but it does generate about 40 gigabases of DNA per lane, so that hundreds of millions of reads. And we do a 2 by 100 base pair reads either end of the library, and then we do an index read to capture that barcode so we can demultiplex the samples into separate bins. So we identify SNPs and indels using alignment programs, and then we compare the variants to the database of SNPs, and increasingly the exon variant server, which is housed here at UW and has become a great resource for rare and common variants. And then we also use the mutation-specific databases like the Breast Cancer Information Core, which has a lot of data from Myriad, or used to have data from Myriad. We identify CNVs by depth of coverage and split reads, which I'll talk about a little later. So our capture size is quite small. It's just over 1 million bases, so 1.1 megabases. This allows deep pooling, as I just mentioned, 96 index samples per lane. But despite this quite high level of pooling, we still get very high coverage, a median of 320x. And when we look specifically at every base that we try and capture, 98.4% of the bases have at least 50 independent reads at each base. So it's pretty high coverage. And we believe that the high coverage definitely improves detection of SNPs and indels, but also allows accurate and robust CNV calling, mutation detection in complex genomic regions, and detection of mosaicism, either somatic or germline. And I'll take you through some of these examples now. Oh, but first, the validation series, sorry. So because these are medically important genes, and this was a new technology, the capturing and the sequencing, we really wanted to make sure that we could accurately identify known mutations. So we started off with 15 mutations from 10 different genes that we'd identify over the years by Sanger sequencing. We did BROCA on them, and then blinded, we found out if we could find the mutations. And we did find all 15. They included nonsense, splice site mutations, and indels up to 19 base pairs. And when we looked at the sequence coverage, we were interested in the balance between mutant and variant reads. And here you can see the balance is pretty good. All of the samples except one are at about a 50-50 ratio, which is what you'd expect for a germline heterozygous mutation. 50% wild type, 50% mutant. The one exception was in a gene called CHEK2, and the mutation was 1100 del C. And I'll come back to that example a little later. So here's some raw output from our sequence data. It looks pretty yucky, as you can see. So we start over here at MUC-YH on chromosome 1 and go across to CHEK2 on chromosome 22. The median coverage, as I said, is about 320x. But there are some pitfalls associated with capturing your regions with this approach. The first is that not all regions are captured as efficiently as others. We have one extreme example, the Peutz-Jeghers gene, STK11 on chromosome 19. And you can see it's only present with about 50 reads. This is an exceptionally GC-rich gene, about 80%. The other problem is that you also pull down gene regions which are highly similar to the gene regions you care about. So P10 is a good example. P10 is on chromosome 10 with its exons and introns, but further up chromosome 10, a couple of megabases away, there's a pseudogene where you have just a very high fidelity copy of the exons. And that's indicated by these spikes here. So the coverage goes up because you're basically pulling down the real gene and the pseudogene. And in fact, we didn't know about the MRE11 pseudogene going into this project, but using this data, we were able to find an uncharacterized pseudogene. And then a similar problem is genes which are segmentally duplicated. So these are very big blocks of homology. And we have 2 genes which are partially segmentally duplicated, CHEK2 on chromosome 22, and then one of the Lynch genes, PMS2 on chromosome 7. And you can see the coverage goes into the thousands here because there are basically many copies of the region you care about at very high copy number throughout the genome. But despite this, through a normalization process, which takes into consideration the CG content and the number of baits at each specific base, you can actually correct it. So this is the same data that's gone through our normalization pipeline. And you see the coverage is pretty tight, around the ratio of 1, the exceptions being the PMS2 pseudogene, STK11, which is pretty noisy because of that low coverage, and then the CHEK2 pseudogene. But in general, I hope you see that we can still make CNV calls once we've normalized the data. So we use 2 approaches to identify CNVs from our sequence data. The first one is a depth of coverage, which we published earlier this year in BMC Genomics. And basically, we normalize all 96 samples that went through a flow cell lane, and then look for outliers that either have a signature of many reads, indicating a potential gain or duplication, and then troughs of reads, which would indicate a potential loss or deletion, indicated in this cartoon here. And this is, I'm going to show you 2 examples of mutations we've recently identified in breast and ovarian cancer patients. So this is ATM, a moderate breast cancer gene. It's got 61 exons indicated by these vertical lines. It spans about 100 kb. And here's the normal coverage going 5' to 3', hovering around 1 until we come to this big dip here, which we validated as a 4 exon deletion of ATM. Here's an example of a duplication that we recently identified. Again, pretty tight distribution of ratio coverage around 1 until we come to the last 2 exons, and it jumps up to a ratio of 1.5. And we validated this as a tandem duplication of 16 kb. So the second approach to CNV detection is actually to use the sequence information in the reads. There are a number of programs available. We use SLOPE, developed by Haley Abel at Wash U. And she developed a split read algorithm that basically looks at the reads and asks, can it map to 2 different locations within a read? So if this is 101 base pair read, can the blue portion map to a different location than the red portion, and consistently to generate a signature of split or broken reads? So we ran this on our BROCA data, and we identified a single exon deletion of BRCA1. And here's the SLOPE output. So here are the normal reads spanning across a region. And here are those split reads, which have their distinct signature. They all start in the same location. And using this location, then we could, which is over here, we could actually pull out the other end of the deletion. So we confirmed it at Sanger level. It's a 522 base pair deletion. The only problem with this approach is it really needs at least one end to be in unique sequence. If both ends of a break are in allele repeats or high copy repeats, then it doesn't work. And in those cases, we rely on the depth of coverage approach. So the second benefit to having this high coverage is mutation detection in complex genomic regions. So CHEK2 is a moderate penetrance breast cancer gene. The real copy of the gene is located on chromosome 22, but then there are partially segmentally duplicated copies on chromosomes 15 and chromosome 16. So there is quite a common mutation amongst Europeans. It's called 1100 del C, and it gives rise to about a 2-fold increased risk of developing breast cancer. And at present, it's identified using a nested PCR amplification. So the first round of amplification is a 10 kb product, starting off in a unique region, which pulls out the 10 kb, the real copy of CHEK22 on chromosome 22. And then you come in with an internal primer and genotype at exon 9, where the 1100 del C mutation is located. So we were interested in determining the sensitivity of mutation detection with BROCA for this mutation. And we ran it on a few samples because it's pretty common. And we found a ratio of mutant to wild type reads of about 81 to 470. So that's about 15%, which mimics what's going on in the genome quite nicely. So the baits that we designed are binding to the mutant copy on 22 and the wild type copy on 22, and then the 2 copies on 15 and 16 and resulting in about a 1 to 5 or 15% ratio. So we're quite happy with this result and suggest that BROCA is sensitive to tough genomic regions like this. The final benefit of having high sequence coverage is the detection of mosaicism. This can be difficult to detect and quantify with standard sequencing approaches. We recently sequenced a patient with a fallopian tube carcinoma, who then went on the same year to develop a retroperitoneal leiomyosarcoma. When we looked at her BROCA data, we found a well-established deleterious mutation in P53. But the ratio reads were not 50-50, which we expected for an inherited germline mutation. So we Sanger sequence validated that, and you can see that the mutant A peak is at quite a bit lower ratio than the wild-type G peak. And we did clonal PCR and confirmed that it's also at about a 15 to 20% ratio. So this confirmed that we could detect mutations of this type, and we believe that the sensitivity can go a lot lower than 20%, but we just haven't come across it in our germline samples yet. We're currently running BROCA on tumor samples, and we're likely to find mutations at a lot lower frequency because of contamination from surrounding normal tissue. So just to summarize, BROCA, our targeted capture and sequencing approach, it seems to be sensitive and specific and accurate for mutation detection in clinically important genes. Pooling of samples with a barcode has definitely increased our sample throughput and reduces the reagent cost per sample. And we believe that a high sequence coverage enables reliable detection of CNVs and mutations in complex genomic regions and mutations that are only present in a minority of cells. So our first clinical application of BROCA was to a series of 360 unselected ovarian cancer patients that had surgery here at UW. Ovarian cancer is the most lethal gynecological malignancy. Unfortunately, most cases are diagnosed at advanced stage of disease when the chance of cure is quite small. Previous studies have shown that about 13 to 15% of ovarian cancer patients carry germline mutations in BRCA1 and BRCA2. And there is a small undefined contribution of the Lynch syndrome genes. But overall, the contribution of other tumor suppressor genes, particularly those involved in breast cancer risk, hasn't been systematically evaluated at present. So the goal of the study was to determine the fraction of ovarian cancer caused by germline mutations in known tumor suppressor genes. So this was a collaboration with Elizabeth Swisher from Gynonc here at UW, and she prospectively enrolled 360 women who underwent primary surgery for either ovarian, fallopian tube, or peritoneal carcinoma in a 10-year period. She enrolled the patients at time of surgery, so there was no survival bias. And because UW is a tertiary care center, it does receive a number of high-risk patients, but she excluded anyone who was referred for surgery for preventative or prophylactic surgery, and also excluded patients who are having surgery for a recurrent carcinoma. So we performed BROCA on 360 patients, the germline DNA of 360 patients, and then looked at mutations. And we focused only on mutations that were considered damaging, well-established damaging mutations. So frameshift, nonsense, splice-like mutations, and in this case, we always validated the RT-PCR level that they led to aberrant splicing. And then the two classes of CNVs, deletions and duplications. For missenses, we only included the well-established ring finger missenses in BRCA1, mutations that are considered damaging in the P53 database. And then we did develop a yeast-based functional assay for missenses and CHEK2. But they were the only three genes where we were confident we could assess function of a missense mutation. So looking across the entire series of 360 patients, 17.5% of them did have a BRCA1 and BRCA2 mutation. When we looked just at the patients that had ovarian cancer, the rate was 16.5%, which is quite similar to the two studies that have been published. The Florida series had a mutation rate of 15%, and then the recent Ontario study had a mutation rate of 13%. So this suggested to us that the patients that we're seeing here at UW at the tertiary center have BRCA1 and BRCA2 carrier frequency similar to the unselected general population. So looking beyond BRCA1 and BRCA2, we identified another 19 mutations for 5% of our patients in 10 other genes. Here are the genes listed here. You can see that we pretty much hit every gene in that Fanconi BRCA pathway. The one exception is ATM. We didn't find a mutation in ATM. So 10 more genes added another 5% of cases with a germline deleterious mutation. I'm just going to go through a few of these genes. The first was the Lynch syndrome genes. We really wanted to know what the contribution to Lynch was for the unselected ovarian cancer population. And it's actually quite small. We only identified 2 patients. And both of them carry germline mutations in MSH6. And when we looked at the histology, both of those were endometrioid ovarian carcinomas, which has been previously associated with Lynch syndrome. We did do immunohistochemistry on the proteins and showed that MSH6 protein was absent. MSH2, MLH1, PMS2 was present, of course. And we did look at the tumor samples for microsatellite instability, this key feature of tumors that lack mismatch repair. And as you can see here, the tumors were unstable compared to their matched blood DNA. We also looked at p53 mutations, and interestingly, the 3 mutations that we identified didn't fit the criteria of Li-Fraumeni syndrome. So the first patient was diagnosed with ovarian cancer at age 64. She had a well-established RR175 cis mutation. And although there's a cancer history on both sides of the family, none of these cancer types are considered Li-Fraumeni spectrum tumors. The second family had a Y226 mutation, and she was diagnosed with breast cancer at age 45 and then a peritoneal carcinoma at age 51. And again, although there's cancer history on both sides, it's older onset and doesn't include the classic sarcomas or early onset leukemias that are typically associated with Li-Fraumeni. So looking at the moderate or intermediate risk breast cancer genes, I think the most interesting one was BRIP1. We found 4 patients that carried germline truncations of BRIP1, 2 frameshift mutations indicated by the yellow lollipops, a nonsense mutation, and a CNV that removed a single exon, a deletion of a single exon, exon 18, which we resolved at the sequence level to be a 2.6 kb deletion. So interestingly, there was a very recent paper from a group in Iceland, and they identified a truncating mutation which is very close to the 2 truncating mutations that we identified. And because this is a founder allele in Iceland, they were able to do a very large case control series to determine the relative risk of having this mutation. And the odds ratio was actually very high. It was an odds ratio of 8 for this truncation in Iceland. So we did identify germline mutations in other genes, in particular the MRE, Nibrin, and RAD50 complex. Mutations in these genes had not previously been associated with ovarian cancer. Also, BARD1 is another gene that hasn't previously been associated with ovarian cancer. So looking at the characteristics of patients with germline mutations, 30% of the women didn't have a prior personal history of breast or a family history of breast or ovarian cancer. And 30% of the women with inherited mutations were diagnosed with a cancer after age 60. So we believe that selecting patients for genetic testing based on family history or young age of diagnosis would miss a third of mutation carriers. So to summarize the ovarian BROCA study, more than a fifth of ovarian carcinomas are associated with germline mutations in at least 12 genes. Mutations in any of these 12 genes, which are in the same pathway, would be predicted to create a tumor phenotype which would be sensitive to PARP inhibitors. We believe that comprehensive genetic testing for inherited ovarian cancer may be warranted for all women, given that the yield is about 20 to 25%, and of course regardless of age of diagnosis or family history. And based on the BRIP1 data, it's possible that genes which seem to predispose to a moderate risk of developing breast cancer may in fact be more important for the development of ovarian cancer. So exome sequencing has become very popular over the last couple of years. It was pioneered here at UW and is a wonderful approach for identifying new genes from various phenotypes. It captures the complete complete coding portion of the genome. It's about 40 megabases to capture the entire genome. As I said, it's highly effective for identifying new genes for simple and now complex genetic disorders. But I'm curious to know whether or not it's suitable for clinical sequencing, particularly when you just care about a very small subset of medically important genes. We haven't done a direct comparison between BROCA and exome sequencing, but there was a recent study from the Cancer Genome Atlas that performed exome sequencing on germline DNA from 316 patients with serous ovarian carcinomas. That was published in Nature in July. Within the Swisher series, there were 242 patients with serous ovarian carcinomas. So this allowed us to compare the mutation frequency between the two approaches and between the two studies with serous carcinoma. So our germline mutation rate within serous was 25%. The TCGA had 14%. We made germline calls in another 5% of our genes, whereas the TCGA didn't make any other germline calls, despite having the full exome sequence. They limited their germline calls to BRCA1 and BRCA2. And I think most interestingly from this comparison is that within mutations, 7% of our mutations were copy number variants, but they didn't make any copy number variant calls with the exome data. So I think the differences in the mutation frequency is likely due to sequence depth. As I mentioned, we have very high sequence depth, but exome sequencing generally bounces around quite a lot. So you will have regions that are poorly captured compared to regions which are very well captured. So the normalization process that we describe, where we have exons and introns, is not going to work as well when you're limited to exons and you're limited to variable capture ability. So I think for current clinical applications, the far higher coverage of targeted sequencing confers greater sensitivity at a much lower cost than exome sequencing. So I wanted to finish with a couple of comments about the patent. So outside of the US, I think it's now easy to do clinical testing of BRCA1 and BRCA2 and all the other breast and ovarian cancer risk genes in a comprehensive, high throughput, and cost-effective way. We cannot offer that for our patients here, but we can continue to improve our research-based test. I'm working with Colin Pritchard and others in laboratory medicine. We now have extended the BROCA design to cover all known breast, ovarian, melanoma, pancreatic, and colon cancer genes, with a goal toward moving toward a single test for broader cancer susceptibility testing. And I'm going to end with a question, the next step. It would be nice to offer BROCA as a reflex test for patients that test wild type at Myriad for BRCA1 and BRCA2, so we can offer a comprehensive base test in the other genes, and also the CMVs, of course. So I'd like to thank Elizabeth Swisher, Anne Thornton, and Ming Lee, and Mary Claire, of course, and also Colin Pritchard, Steve Salaponte, Christy Smith, and Dr. Tate, who have been working closely with us on the HiSeq and developing ColoSeq. Thank you.
Pretty exciting stuff. It sounded like you were very conservative intentionally in deciding on classes of mutations you try to pursue in your analysis. But did you look, how much did you look at the less conservative same distance mutations outside of the 3 genes that you included? Did you, how many more women with otherwise no genetic lesion did you recruit?
We did look at other missense mutations, and we used prediction tools like SIFT and Polyphen, but we found those actually to be quite unreliable. And I think having the Lynch genes was particularly informative. So if you have a mutation in the Lynch gene, the tumor should have microsatellite instability. So we found a series of, I think, 12 missense mutations in the Lynch genes that looked really damaging by Polyphen and SIFT. But when we looked in the tumors, they didn't have microsatellite stability. So we classified them as benign, rare and benign, based on that test. But we can't do that for the other genes because there isn't a simple assay.
So have you given any thought to looking at not obviously the whole genome, but like enhancer promoter regions that are upstream or downstream of these known genes?
We limited it to 10 kb. In the very first design, we actually went 100 kb beyond BRCA1, because we really believed there was something going on the upstream region, but it was too much data to look at, really, so we decided to limit to 10 kb. Hopefully, the enhancer prediction will improve, and we can then go back and look at the mutations that we have and see if they do affect expression.
Could I follow up on that question, that the capture strategy by hybridization, you had 120 base pair capture probes that covered the entire exon sequence of interest, right? And when you went 10 kb, you covered all that 10 kb, or you just looked for particular regions within?
No, we blanketed the whole region, just taking out repeats. So if the gene spans 100 kb, we'll capture the entire 100 kb minus the repeats, and that varies. It's usually 50, 40%.
So did you learn anything through this process of hybridization capture about maybe designing the future versions to optimize capture? Because that slide you spent some time on where you get differential coverage.
That we are now trying a boosted bait strategy, particularly for the STK11. They're going to manufacture more baits of that region first, so that should improve the coverage. For the segmentally and pseudogene regions, we can't distinguish between the real and them, so we just have to keep capturing the regions we don't care about.
It's a lower frequency condition, but have you looked at the operation of the BROCA assay in male breast cancer and what you end up seeing?
We haven't, no. At the moment, we're focusing on Marie Claire's series of high-risk families and unselected patients from various other cohorts.
Many of those families did look like some of them had male breast cancer.
They did, yeah. PALB2 is considered the second big hit for male breast cancer after BRCA2.
What types of samples are you using for the BRCA testing? Is it paraffin embedded tissue or?
We started off with germline DNA extracted from blood, and we've done frozen tumors, and we've done a very small series of paraffin-embedded tumors, and it works well. We can't make copy number calls very accurately because the DNA is pretty degraded, but we can find small mutations with a high accuracy from paraffin-embedded tissue.
Ultimately, what proportion of hereditary cancer do you think you're missing in the BROCA set?
You mean in terms of new genes?
Yeah, so what percentage of breast cancers, hereditary breast cancers that lie outside the BROCA set? Ultimately. Even if you had like the upstream region you could call correctly.
Well, based on our initial study of high-risk families, about a third of them don't have mutations in these 21 genes. So they're now going through an exome sequencing pipeline to identify new genes. And it's very likely there are additional genes that we're not sequencing in a high-throughput way at the moment. But then once we identify those, we'll update our design to capture those genes.
The two patients with endometrioid, ovarian, and the Lynch mutations, did they have concurrent endometrial cancer in their uterus?
I don't think so, but I'm not sure. All right. Yeah.
That I should know the answer to. Great talk. What do you think is the minimum depth of coverage that you would feel comfortable with for accurate CNV calling in a clinical assay?
That's a good question. I think it's about 100x. Yeah.
And then the clinical exome sequencing that's being offered by Ambry Genetics, I think they're advertising about 60x.
I think it might be more than that.
Oh, more than that.
Maybe, yeah.
But not quite enough.
No, I did ask them if they could make CNV calls, and they're not even going to attempt it based on their coverage. But you know, as capacity increases, I think coverage will go up, and I know the ICLA lab are close to working out CNV calls from their exome data, so I think it will come through eventually.
You mentioned 12 mutations that when found, led to the prediction that those malignancies would be susceptible to PARP inhibitors. And so I'm wondering, is there research going on to look for mutations in genes or gene products that are PARP substrates, and or are there other chemotherapeutic agents that might be useful directed at some of those targets of PARP, or is that just such a big ocean of proteins that it's not?
No, I think it's a great idea, but I'm not aware of anyone doing it. But I mean, I think the future is to target pathways that you know are involved in particular cancer subtypes.